Okay, so we'll start now formally and we'll be recording you as well. So let me just introduce our next speaker, Professor Guillaume Mata. I hope I'm pronouncing your name almost correctly.
Guillaume.
Yeah, Guillaume. Okay.
Yeah.
So Guillaume is a biologist, like many people in the audience. So Guillaume is interested in evolutionary dynamics in non-equilibrium populations, non-equilibrium, for example, due to demography. And he is currently a faculty at ISEM in Montpellier in France. And he will be telling us interesting questions and some answers in evolutionary rescue. What do you, Guillaume?
Hello, so thank you very much for inviting me to this lecture. So yeah, I'm going to do three lectures of an hour and a half on the theme of evolutionary rescue. Evolutionary rescue is a process by which a population that is doomed to extinction will avoid extinction through some genetic change. I'm looking for my notes. That's the thing I didn't. Okay, they're here. Sorry. So it's really, in essence, something that has demography and evolution by definition, because the definition of the issue is demographic. There would be an extinction of the population in the absence of genetic change. And the evolution aspect is also in the definition by the fact that we call it evolutionary rescue. There are other ways that a population may go down and then up again in size that are not evolutionary, that they don't involve genetic changes in its composition. But evolutionary rescue is really focused on this aspect. I'm going to show you quite most of the class is, I didn't do slides. Most of the class is going to be by writing on my tablet, because I saw that Kavita was doing this and I thought it was great. So I hope to try to do the same. So I'm going to kind of switch between different shared screens. Mostly it will be the whiteboard, and then I will share screen a little bit with some examples of paper and some Mathematica notebook. The reason is not to do a practical on Mathematica. It's just to illustrate, especially for those who, like me, have had trouble in their training doing math and calculations. I always had the numbers wrong all the time. And then I realized that partly some tools can help you do that automatically. So I think I personally, in my practice, think it's quite useful to see that some things can be computed by a computer. I mean, math things, analytic, not numerical. And also it helps focus on the modeling instead of the math, because modeling is not math. Modeling is going from a reality, a view we have of it, and then the simplification we make of it. And then we go into the realm of math and we can use the tools of math. So maybe I will start by an overview of the things I hope to talk about. So this is going to be my whiteboard. So today I hope to talk about basic, it's horrible, it's not working. Basic evolutionary rescue in the simplest situation. I will call that ER because it goes faster. I will deal first with a tool, and it's going to be a lot about tools first. S. And a very important tool in evolutionary rescue is the Poisson distribution and process. So I will do a little bit of, for some, revision, for others, maybe not revision, but I'd rather have those things clarified, and a bit of using Mathematica to compute those things. Then I will use that And we will try to derive ER in simple scenarios. And we will go from the simplest to hopefully more complex cases. But that will be for tomorrow. Then the next lecture, I hope, is about basically adding first, more complex genetic basis. You will see today that we are going to consider rescue with a very simple demography and with a very simple evolution. And also connection between stress and rescue. Because in many cases, we have control over the stress level. We hope, or we can measure the stress level, and we hope to use that or to predict things from, sorry, I'm new to this whiteboard, so I'm going to do a bit of mistakes sometimes. In many cases, we either control the stress level we impose, for example, with a drug dose or things like that, or we can measure the stress level. For example, in natura, we can measure the kind of stresses that are occurring on the populations and have an idea of what it does. So we need something in the rescue theory that connects this level, this quantifiable level of stress with the ER process. Alors, and a third lecture will be roughly also this, and extensions to even more, I would think, more complex demography mostly, although I will remain, some extension to more complex demography. Because of course, there are assumptions we have to make on demography and on evolution to do an evolutionary rescue model. So ER, so what is it? I will write the definition again. Process, it's a process where a population decays and it leads extinctions. Is it okay? You know this when I do the little O, is it fine? If you cut me, of course, when people don't see what I'm writing or understand what I'm writing. That's the principle, I think, here. It will lead extinction, certainly. And evolution, meaning change in the genetic composition of the population, allows to avoid decay, or at least, put it otherwise, to avoid extinction. So this is really the two things. So we cannot define evolutionary rescue if there is not initially a risk of extinction. So that means that a lot of the population genetics that we are used to, which are based on, say, the Wright-Fisher model, etc., they are in stable populations. So we go from stable populations to non-stable, non-equilibrium demography. We have to go to non-equilibrium demography. That's the first thing. Another aspect is that it happens over time scales that are short, that are the demographic time scale. So that means that we must also have non-equilibrium evolution. We cannot use, without thinking about it at least, all theories that are based on a stationary state process, mutation selection balance, traveling waves at stationary speed. All those tools, we cannot use them a priori because they require, to be valid, they require a burnout time where we let time evolve. And after some time, that can be quite long, we observe these stationary regimes. So that means that mutation selection balance, sorry, my pen does weird things. So it means mutation selection balance, which I will write MSB, mut cell balance. I think you've seen that concept when mutation and selection balance each other to maintain some diversity at some stationary level. We cannot use them. Traveling waves of the adaptation, which occur or not depending on the assumptions we make, are also stationary regimes, but they are steady state regimes where fitness increases, so it's not an equilibrium, but it increases at a stationary pace, and there is an equilibrium of the variance of the system. So these we can't use either. So these are important tools in population genetics applied to demography, to fitness, and we can't use them. Similarly, adaptive dynamics in the sense, sorry, there's really a problem with this. I hope it's not going to be happening all the time. Adaptive dynamics, I don't know if you've heard about it, but they are really repeated evolutionary games. And the time scale is the replacement of a population by a strategy that replaces it to become the new dominant strategy, and then another strategy, and then another strategy. In those approaches, we are following iterated games, like the prisoner's dilemma games, but they are iterated depending on the strategy, and then the next strategy is just a slight difference away from the first strategy, and by local analysis, you can see the invasion of a new strategy into the dominant one. This is very useful if you want to deal with especially interaction and frequency-dependent selection, but it deals with first a timescale that is not defined directly at the demographic level. It's the timescale of invasion of a strategy by another. So it's a very long evolutionary timescale, and it's iterated. And so you cannot connect it to a direct demography of per hour rates. So you cannot use that either. So it means that we must use our classic population genetics, and it's really stochastic demography. The thing is that there is a connection between this demography and evolution. So this connection, there is a cross here. This connection happens through fitness. Fitness here is going to be what is, fitness is not something you can just invent and say, I'm using a proxy for it because it has no definition. Something you can hear sometimes. It has a very clear definition, at least that's my opinion. And when you use something else, it's a proxy for fitness, but you cannot say that you can use any proxy and it doesn't matter. Why did my word disappear? I don't know, sorry. So fitness here is going to be the expected change in log copy number of a genotype. So it means that fitness is, of course, not reproduction, it's not survival, it's none of these. It's the expected change in copy number. Reproduction is the number of offspring of an individual. If you have a model with, say, a discrete time and fecundity and survival, so say plants, annual plants that have a given probability of surviving over 1 year, and then a fecundity in terms of number of seeds they produce, a given plant will have a given reproduction, it will have a given survival, 0 or 1, but its fitness cannot be defined at the scale of an individual. It's defined necessarily at the scale of an expected process over ideally infinitely many copies of the genotype considered. And that means that with sexual, it's much more complicated because we have to define fitness at the scale where the genotype doesn't change. So usually we define it at the scale of the locus or of the site. Here, we're going to deal with two simplifications, most of it. We're going to deal with asexuals. This is simplifying in our case. In some cases, the population genetics of asexual is quite messy. It is in our case too, but it's also simplifying by the definition of a genotype that doesn't move except by mutation. And we're going to deal with density-independent demography. So why do that? You could say that we do that because it's comfortable. It's not a proper reason, obviously. The reason is that when you consider a demography of a population that is decaying, usually if I draw, so this is a draw I will do a lot, I guess. I will draw log n, and that's a log pop size. So that's going to be log pop size versus time. If I do a draw of a logistic equation that has density dependence, it goes like this. But if I look now instead at, oh, that's terrible. Okay. I didn't see that coming, I'm sorry. Okay, so I don't know what's happened, but okay. If I look at a population that is now going down, typically, say it is stable, for example, and then it's going down. And there is a stress, the environment changes at that point. Stress is imposed here. A lot of what's happening is happening at population size that are way below the density dependent level. It's not completely true because of course here, some things are happening at an area of pop size where there is density dependence. But most of the process happens in the process where the population becomes smaller and smaller. So it means what? It means that it's not crazy to ignore density dependence. These areas of population are roughly density independent. This neglects what is called alley effect, which I don't know if you've heard about, which are things that happen, density dependent effects happening here at low population size. Alley effects can be of many types. The typical one is when you consider that how does a population reproduce when it's in very, very low density. If it is a sexual, obviously it's very complicated to find a mate in the middle of the desert. So that's an alley effect that is typically assumed. There may be other things that happen to organisms that are not sexual and that do not require a partner, but there are less of them. Here, what by bias I have in mind is mostly bacteria, viruses, which are in majority asexual. They are not asexual at all, but they are doing most of their biomass increase in reproduction as asexuals. And they have, we can assume they have little alley effect. We can also measure dynamics of extinction and see that in log scale, they look like straight lines. So it means that it's not crazy to consider a density independent process. So really, this density independence, it depends on log linear decay. So it's a measurable question. You can measure it and see if you're observing log linear decay. So that's going to be about the extinction process. Having density independence is very, very handy. The reason is that while we can do density dependent models quite easily when we are dealing with deterministic modeling, say the logistic, etc., it's much more complicated to do stochastic process with interactions between lineages. And so a lot of things have been derived and obtained in contexts where there is no interaction between different cells, for example, or different lineages. That means that asexuals means no interaction because there's no recombination going on between different strains. So no genetic interaction. And density independent growth means no demographic interaction. We will see that the problem is already complicated enough in this context. Also, you can mix, we'll see that in the end, you can mix some, say, density-dependent growth with the density-independent process in some extensions. Typically, you consider that your non-mutants are deterministic and you can play with them with density-independent demography there. And your mutants, you consider them at low size and they are not acting on the system. They are just undergoing their own fate. So those two assumptions, we're going to deal with sexuals at one point, but most of what I will talk about is density-independent demography and asexuals. Finally, the other important simplification I will do in most of what I will do is a constant stress. So that's very important because there are many, many situations where we can expect not to have this. But for starters, it's very important to understand what happened when you have a shift between, oh, what is it doing? Sorry. I can't find my slides back. I don't know what's happening. Okay. I don't know what's happening. It's doing things that I can't control. Anyway, you have.
Guillaume, is it possible to save what you're doing once in a while, like, in Word?
I'm trying to. Normally it should, and normally it should do slides, and it doesn't. So I don't know what's happening, really. But I'm going to prepare slides in advance, and so that they will exist already in the system. So here you have time. Typically, time 0 for me will be the onset of stress. From then on, the wild type, the dominant population decays. As is at the onset of stress, the population as is would decay. And before that, this is a permissive environment. Meaning, environment, I will write it this way. Stress. Meaning by permissive, meaning it may be stable population, or it may be an increasing population in a preculture, for example. But anyway, things are growing here. And things are decaying here. And stress is going to be constant. So the conditions are stable during the whole process. Of course, there are conditions, for example, with treatments, where stress goes like this. Initially, there's nothing, the dose goes up, and it goes back down. It's actually very hard, I found it in my experiments at least, very hard to produce a stable condition with an antibiotic, for example, because the antibiotic takes time to act anyway, even if you put a fixed dose and that there's no loss of the antibiotic in the system. So we'll deal with differences like this later on, but to understand ER, you need to understand what happens in just one change in condition from previous to next. So a typical ER process will be like this. We have a demography of type AI, and we assume that I use prime for derivative with respect to time. That is, demography is MI, NI of T. And that's NI is number of type I. This thing, this would be fitness, and more precisely, Malthusian fitness, meaning the growth rate of the copy number. In fact, this simple view is not the correct one. What we have in fact said, and what is said in the models, it's not, is that the change dn, the change dn I over ni dt, given the current state ni, equals mi. So it means that fitness is the expected change in log ni, given ni. That may seem like details, but it's not, because otherwise people mix drift and selection. To me, genetic drift is all about the non-heritable variance in reproductive success, and selection is the part that is heritable in reproductive success. And in stochastic demography, we can model a demographic process by having an expected change and some noise in demography. So clarifying this allows to clarify what is what is fitness and what is something that would contribute to drift in the process. Anyway, here for the moment, to clarify, to simplify, I will just keep things by saying when I use this, I mean, in fact, the expected number of individuals of type I, but I will simplify by not putting the expectation of every time. It so happens that when we have density independent demography like this, why do I say it's density independent? Because this thing means that if you know derivative, ni prime of t over ni equals mi, and that thing is the derivative with respect to time of log ni. So that means that in log scale, your population goes as a straight line down or up. And that's a definition of density independent growth. And because there is no interaction, this happens to all types because they are not affecting the other types. So that's very important because it means that we can deal with every type independently of the others. And that simplifies things a lot. The other important thing is that when we measure this kind of exponential growth, we have access by the straight line to the fitnesses of the individuals, but we have access to them by taking expectations over large population numbers. And actually, the model on the expected size actually grows exponentially. It's not the actual size that goes exponentially. It cannot, because the actual size is an integer, and an exponential growth is a real number. So you cannot have a real number describe a count of bacteria. So in fact, the very fact that we have exponential growth or decay is in fact should make you remember that it's an expectation that we're following, not an actual count. So the connection between demography and evolution is going to be through this. If we have ni prime of t equals mi ni, then you look at the total population size of the population. Big N equals sum of ni over I. So several types together. Then you have that n prime of t equals the sum of the ni prime, which is the sum of the mi ni. And that thing I can rewrite by saying it's the sum of the ni over big N times the sum of the, the sum of the yeah, it's a bit new to me. The sum of ni over big N, which is the proportion of type I, times the Malthusian fitness it has, this thing, times big N, right? I just put big N, I just have put big N here and here, okay? But what is this thing? This thing here is the sum of the frequencies, pi, mi, multiplied by I. So this is the Malthusian fitness mean, average Malthusian fitness. So if you have several types growing density independently, you have the total size grows as the mean Malthusian fitness, which depends on time because of the frequency is changing with time, even if the fitnesses are intrinsic properties of the genotypes, times big N. So now you have a population which growth rate in log scale is given by the mean Malthusian fitness. This is the mean Malthusian fitness. So with this, now we can do the connection between it's a mess. With this, we can connect the evolutionary and the demographic process. Why? Let's plot the pop size in log, the total pop size versus time. Where time zero is the onset of stress. And below it, we're going to plot time versus the mean Malthusian fitness. We have seen that m prime over n of t equals m bar, which is really the derivative of log n. So that's the slope m bar at every time is the slope of log n. And we start with a decaying population. That means that its mean fitness, absolute mean fitness, is negative at some value r0, which is less than 0. And this value is the slope here, the slope of the log scale decay. And if nothing happens, no evolution, we have a population that goes extinct. Now, a first scenario may be that there is evolution and some mutant invades or something happens to the fitness so that it changes and increases, because fitness by selection can only increase. Then the demography will change here. The slope will not be 0, it will be less and less and less. And in the end, it will be something like that with a new slope, but a slope that is still less than 0. So this is, there's an evolutionary process, there's a demographic process, but there's no rescue because the evolutionary process did not produce a fit enough end result. We have another possibility, which is that there is a change that happens in mean fitness due to whatever changes the mean fitness. And this change happens early enough and to a high level enough that before extinction, we have a change in the demography. We go to a positive mean fitness in the end. So that would be R plus, for example, and it would be given by the ultimate slope of our population once it happened. This is now an evolutionary rescue. What is important is that also you can have a population that if it was in an infinite size, would have gone this way in fitness, like this, and then a wonderful, very high fitness. It would have reached a very high fitness by its process of evolution alone, without demography with it. But this happens too late. This is way too late. The population is dead by then. There's no rescue. Although the ultimate state, the steady state, does not give us any information, because the ultimate evolutionary state is not relevant to rescue. And even if the ultimate evolutionary state would have been very fit, it's not enough. It needs to be fast enough. So rescue is really about computing a race. Why do I have a blue thing in the middle? I have, oh, sorry, it's horrible. I'm sorry, it's horrible. Anyway, I had finished my slide. It's a race between extinction and adaptation. And as it's not, as it's a race, it's all about non-stationary problems, dynamics. I am scared to use the, whenever I use the eraser, I'm erasing everything. So I won't use the eraser anymore. So the key, of course, is to know two things. To model the change in the mean fitness, this thing, and to model how this affects the demography. So that's evolution, that's demography. For demography, it's quite simple because we have seen that the change in log n is equal to the mean fitness. So in our simple context with density independent growth, we don't need to worry about how this evolution will involve things for the demography. Basically, if we manage to correctly model the dynamics of M bar, we will have our evolutionary rescue theory that should work. So now the question is how we model this. So this is about changes in mean fitness. So all the evolutionary forces may affect the system. So you have mutation, you have selection, you have recombination, sex, because there is also segregation, you have migration, and you have drift. I will consider isolated populations. So migration, I don't consider it. If you're interested, I can talk about it in questions or point to papers about it. I will mostly not consider sexuals. I will do a quick overview of one historical and important sexual mode. But what remains now that we cannot remove is necessarily we will have to deal with mutation because in asexuals, it's the only source of variation. And we have seen that we need genetic change. So we need variations. Selection, we will have to deal with it because it just is a consequence of demography. If you have mutation without any selective effect, it means that you have changes that do not change the demographic rates, so they cannot produce rescue. And drift could be avoided. Some models of rescue are deterministic, but most of them have inherently drift in them. In fact, there is more than drift. There is the whole stochastic demography. The reason is that even with some process that has all the parameters set, if I do replicates of this model, I will have sometimes this, sometimes this, sometimes this, sometimes this. And this is the variance introduced, the stochastic variance introduced by the demographic and mutational processes. And that thing, of course, will imply that you have sometimes this, sometimes this, sometimes this, etc. So you need to handle the stochastic process. The reason is that most of the interesting rescue problems happen when typically this is not too large. So at the minimum size, we have a small population. So we cannot say we're going to be all deterministic all along, because if we are in conditions where everything is deterministic all along, basically there's no issue of rescue. There's going to be a visible decay and regrowth, but there won't be an extinction happening, and there's no question whether it's probable or not, likely or not. So, oh, it doesn't come out. Right, okay, I don't know how it works. It's a
Can
yeah,
I ask a question? So when you said that we're basically looking at mutation as a necessary feature here, are we then not considering rescue via standing genetic variation?
Yeah, but this standing genetic variation has been produced by mutation.
Right, but existing.
Yes, yeah, we are.
Past,
definitely
right?
We are.
Okay,
yeah,
so we are considering.
Because the processes that we have to put in the soup are mutation, selection, and drift. We don't put recombination and sex because we're dealing with asexuals, and we don't put migration because we are considering an isolated system. So it means that we put mutation, and standing variance just happens through mutation in an asexual. So we are definitely going to do mutation in standing variance.
Okay, I mean, I think of them as two different things, no? But anyway, maybe we'll come to it later. I'll wait until you.
The standing variance and the de novo, yeah, it's important and it's different, but it still is the same organism doing mutations.
Okay. Just
the frequency they reach that change, their dynamics, their fitness effect. So. Vous avez écrit le maximal de tableau. Okay. It's unbearable, this system. So I cannot create any new board. Okay. I'm going to just erase my graph if I can. Okay. I can erase everything apparently, so I'll erase everything. Now, there is another thing that we cannot avoid is a change in the environment. Because there is a past environment and, say, a future environment. This environment has stress in it. This environment is permissive. So there is things happening in the system in the past, and they will be corresponding to standing, they will be producing standing, I don't dare to use the eraser, standing variance. And there will be things that can happen that will produce de novo, it's called de novo mutations. And rescue may happen from two different sources. Now I'm going to show it as a log n graph, but I'm going to show the wild type. It's going to decay and say in the past it was at stable size. And the rescue mutants may have been produced in the past and wandered about at low frequency. And then there's a change in the environment. And in that new environment, it can grow. Because it's not hindered by the size of the others. So there is, in fact, density dependence in the models of rescue, in many models of rescue, but it's in the past to keep things stable. You cannot keep a population stable without density dependence. So there is density dependence in those models, but it's in the past. And you can have also, so this will be called the standing variance rescue because it comes from the past. It didn't appear, it's a mutation that appeared in the past before the stress. And then there is a mutation that may arise produced by the decaying population during the stress. So then it's like this, and it will rise. And this is going to be a DNER, so de novo. De novo here and standing here. And a lot of rescue theory, at least in asexuals, has focused about this portion of de novo versus standing. I'm not sure it's so important in the end, but it's interesting to look at it. What to me is critical is to know how a strong stress or a low stress will affect the probability that rescue occurs, whatever its origin. But what's important is that you model things very differently. In modeling, it's important because you model things very differently. The process of occurrence of the standing variance and the process of occurrence of the de novo is very different. In the first one, you have the past environment, whereas in the new one, you have another environment and a decaying population. So the critical thing is to be able to model the production of these. And the production of it is via mutation for asexual. There are two things to be an ER. You need to be a mutant with a growth rate R that is positive in the new environment. Which I call a resistant R. It's not a rescue. A resistant is not a rescue, just like a beneficial mutation is not a fixed mutation. The reason is that you need also to be an ER to avoid stochastic loss when rare. And you are rare. Why you are rare? Because if you are not rare, we don't even see a rescue. What we see when there's plenty of resistance mutations around is something like this. We almost see nothing. So for rescue to be visible and relevant, it has to be conditions where resistant mutations are rare enough that we don't see them occurring all the time. Otherwise, this is a population that is undergoing a change in its environment and has way enough variants available, way enough numbers available that you don't almost see anything in its demography. You see genetic changes, but without demographic trace signal. So what do I say by loss when rare? Well, this is really like what you saw, I guess, in Kavita's class for fixation priority. When you arise in single copy or in a few copies, a lot of your copies are just lost. They go up and down because of your trends. Your deterministic trend is to go up, but the noise, demographic noise around it, makes you reach the absorbing state that is zero. So you get lost. And in your previous course, for example, you have the fixation priority equals twice the selection coefficient. We have comparable things here, but they are not in terms of selection coefficient, they are in terms of growth rate, absolute growth rate. So it is critical to model the mutation process and to model it not as a deterministic process, but as a stochastic process, because these happen rarely. These mutants, they are rare. Otherwise, ER is not relevant to study. So we cannot remove the stochastic mutation unless we are dealing with an irrelevant biological situation. So if I can do that, no, apparently I can't. Okay. The key is to model the mutation process. And to do that, the main tool is the Poisson process.
So I have one simple question.
Yeah.
Yes, so in the so-called permissive phase, why can't I think of population in mutation selection balance? And
we will do this one. We will do the
Okay.
mutation selection balance initial state. It's the first example we will do. That does not answer your question, maybe, sorry.
No, I was just wondering that, you said stochastic mutants, but I would have thought perhaps first I will model that phase as just, mutation selection balance, and then let the new mutations be there. In fact, standing genetic variation, just deterministic one.
Yeah. So I will tell you the two main examples I hope to do today. The first one is this. You go for a very, very long time. This is time. This is onset of stress. So this is 0. You go for a very, very long time at a very, very large population size, or you go for a very, very long time at a finite but large population size. So in that case, you're in mutation selection balance, and you can model it deterministically because it's a huge population. In that case, you're at mutation selection drift balance, and you can model it using equilibrium stochastic modeling and diffusion. In the two cases, after that, you consider that you have a change in the environment, and you also consider that you have a drop. The reason is that if I don't put a drop to my infinite population, rescue is irrelevant. It will do this. I'm sure, because there's an infinite number of guys. With an infinite population initially, there is no reason to study rescue. I mean, we can, but there's no issue of dying out or not. It's certain you will survive. But what happens in experiments...
So sorry. My question was about, I thought you are talking about stochasticity in the permissive phase. I thought that Can't I avoid that and just talk about?
You can't really avoid it, because if you want to avoid it, you need an infinite population, and an infinite population has no issue with rescue. So there is stochasticity by two ways. In the infinite population, it's the bottleneck here that puts stochasticity in the number of types you get at the onset of stress. So there is a bottleneck here.
Yes.
And in this one, there is stochasticity inherent in the drift of the drift balance. But you have some stochasticity, which means there is a distribution of the number of resistant types in your base population. Because if there is an infinite number of types of resistant types in your base population, there is a probability of rescue of 1. So there's no reason to study it. Okay. So you need necessarily the standing variance types must be distributed.
I see.
Otherwise, it's impossible. But it doesn't mean that you cannot model the frequency of those types deterministically. But then you sample a finite number of those in this frequency, and you get again a Poisson. So we're going to get Poisson everywhere.
All right.
But if you don't get a stochastic number of individuals, it means, I think, it means somehow you have set a purely deterministic process in the permissive phase. And it means that because it's deterministic, you have an infinite, rigorously, you have an infinite number of individuals. And so whatever the frequency you put in that, there's an infinite number of resistance. So mathematically, you have to put a finiteness somewhere. But in the basic rescue model, we have a deterministic frequency at mutation selection balance in our huge infinite population. And then after the bottleneck, we just sample from that. And sometimes we get no one, sometimes we get one guy, it goes up. You see?
Yeah.
In the mutation selection drift balance, it's different. We're like this. So we have the states oscillate between 0 and something polymorphic. So it's stochastic in a sense before any bottleneck. You don't do any bottleneck. It so happens that the two models converge to the same thing. They converge to a Poisson number of mutations present at the onset of your stress. So that's going to be the standing variance part of it. And then there will be a de novo, which also gives you a Poisson number.
No, so if I understand you correctly, you're saying that I could have either mutation selection balance or mutation selection drift balance in the permissive phase. But when I, at the onset of stress, I will sample these mutants stochastically. It will be some random sampling, because of which, in that sense, the drift can enter. I'm not.
Well, I don't tend to think about drift, because drift is really thought of in stable populations. I mean, even the model itself is the Wright-Fisher model is at a stable population, and when population size changes, we try to force it into different stable populations. So it's really stochastic demography, if you want. But anyway, the bottleneck here there is, in this first scenario, we have to have a bottleneck. Otherwise, we're starting from an infinite population.
Sure.
So this bottleneck creates stochasticity. Although we were able to model the frequency, the past state before the onset of stress, we could model it deterministically. But we had a bottleneck, because if we don't, we have an infinite probability of rescue of 1. We can, of course, retrieve the result. When we say the bottleneck is very small, we retrieve the result and we see probability of rescue is 1. In the other one here, you don't need a bottleneck, because you're already at a finite size, and you say that it's a finite size, and then things change and you switch to decay. So there, you don't have a bottleneck, you already have a stochastic process here, which is the fact that a first dynamics will be this one, for example, the one in red here, but another dynamics of the drift mutation selection drift balance would be another one. And so there are as many dynamics as there are replicate populations that we consider. And as this is, maybe I should have said that to the students, because but as this is a stochastic problem, all that we can say about it is about what happens when you do a lot of replicates of the same evolutionary decay experiment. So you put replicates of populations that have undergone the past in permissive conditions, and then a change in the environment, and you don't look at one of those. You look at a set, many of those, and you estimate the likelihood, the probability that the population will get extinct versus survive. So all this depends a lot on some properties of the mutation process, which is modeled as the Poisson process. And the reason why we model it as the Poisson process, I've often seen it in my class when I was a student. People were saying, we model mutation as a Poisson process, or we model it as a deterministic process. And I didn't know where it came from. Why the Poisson? A bit like in quantitative genetics or in statistics, you model the variance in the error or the distribution of the error as a Gaussian, or you model the breeding values of your trait by a Gaussian. And sometimes it's explained why, a bit more, I guess, because there's the magic of the central limit theorem. But it's not explained so much with the Poisson, while the Poisson has, to me, it's a bit another central limit theorem. It's the law of rare events when the Gaussian is the law of frequent events. And so we're going to see this very quickly. And it's not an issue if I have no time to go on with rescue after that, because I will do it at the next lecture, and that's fine. I hope to do one rescue example, so I have to go fast enough that say I will allow myself a quarter hour on the Poisson and then a quarter hour on one rescue model. Okay, but you can cut me. Huh? So I will change my share screen. I will leave this one. Because I want to illustrate it using Mathematica, because it allows me to show you some distributions, etc., and some computations. This notebook, I will put it on the Slack afterwards, so you will have access to it if you want to read it later. So it's here. Okay, so normally you should see a notebook where it's written Poisson distribution. Do you?
Yes.
Yeah, I think you see it because I see my own notebook on my own connection. So the Poisson distribution, which I denote here, for example, P of lambda, is a distribution with a single parameter. It's a discrete distribution. So it's made for modeling counts, which is important because we are going to count a number of mutations, a number of sites mutating on the genome, a number of events of mutation over time. It's all about being a number. So it makes sense that it's a discrete distribution. It's with values in the positive integers because it's a count. At least that makes it modelable. We can model it as a count. What does the Poisson distribution look like? So to know what it looks like, typically, so I've defined here a function which is just saying P is a Poisson distribution. So that, and then I can see what is the PDF. So the PDF, I hope you know what it is. If you don't, ask me. It's the probability density function. In the case of a discrete distribution, the PDF has a very simple interpretation. It's the probability that your variable is equal to a given number, 0, 1, 2, 3, 4. So this is a graph that goes up and down, and it gives you the probability of each value, possible value, from 0 to infinity. Here you have an example in the graphs below. So don't try to understand the code itself. It's just to see that we can do some code doing this. This is the PDF, so it depends on the value you're looking at. And sorry, I did it wrong here. It's a probability of x equals k, so I should have written it with a k and not an x. It's the habit of working with continuous distributions. So this distribution has the probability has a simple formula. It depends on the parameter, the one parameter of the Poisson, and lambda, and on k. And here is what it looks like. When lambda is, say, 0.5, you have something that goes from very likely of having 0, counting 0, and then a bit less likely you count 1 value. Your number is 1, your count is 2 is a lower value, and it goes down and down. And as you increase the parameter lambda, you have a distribution that looks more like, do you see my mouse here? That looks more like a gamma. You have some excess of counts that are more and more frequent. And as you go with larger values of your parameter, it starts to look like a bell-shaped, like a Gaussian, if you wanted, in continuous space. The properties of this is that basically the parameter lambda, the single parameter of the distribution, is the expectation. So that's why when lambda is small, your expectation is around 0.5. So you expect to have a lot of cases where it's 0 and some cases where it's 1. When your parameter is 10, like it's the green curve, your expected number of counts is 10. So you have a lot of them around this, and then it falls down. The Poisson has a particular property that the variance is equal to its expectation. That's used when you want to test for a Poisson distribution in data. Usually you do a fit of your data. And an important property we will use is that if you know, it has a very simple expression for the probability of counting 0. And this is just this, exponential minus lambda. And that will be very important. Why? Because we're going to be counting rescue mutations. And when we're going to be doing everything to predict the probability of rescue, we're going to always do the probability of getting extinct and say, if you're not extinct, you are rescued. The reason to do that is that we are considering density independent models and asexual models, so without interaction. And so it means that if you don't get extinct, you have to explode. You're either getting extinct or exploding. Of course, you don't explode to infinity, you stabilize to some logistic growth, but everything is decided in your low population size problem. So that's why it's density independent. So the probability of a Poisson number being 0 is going to be very important because it's going to be the probability that we have no rescue mutation, so that we are getting extinct. That's why I will call this. So in Mathematica, you can write those. It's basically a Mathematica is a software to do analytical mathematics. It does it for you, and it has a lot of things in database and knowledge of things about, like I can say, for example, probability that x equals 0 when x is distributed as a Poisson with parameter lambda. Now, a crucial tool in stochastic demography and in probability is a tool that is not studied almost, at least I didn't have it as a student in biology. And I think it's a pity because although it's a kind of an exotic tool, it's in fact simple once you accept to use it. And it's called moment generating functions. So what is a moment generating function? It's a thing that characterizes a distribution, but in a different way from the histogram. The histogram tells you the probability of each event happening. It's more intuitive. The moment generating function is an expectation taken on the variable that depends on some variable, some coefficient, some dummy variable, we say Z. And it's really the same kind of thing as the Fourier transform. For those who have done Fourier transform, Laplace transform, all those transforms. So for those in physics, you are used to the Fourier transform, which puts you in a realm that is not the real realm. And you can study it in this new space, and you have information and you get back to the space of the real process. Well, the moment generating function is really a transform that does that for the space of probability distributions. So what is the MGF? So moment generating function of a distribution, whatever it is something for a given, it characterizes a distribution as much as a histogram, as much as a PDF. It has a variable, a dummy variable Z, that has no meaning directly. And it's the expectation over this distribution of exponential of Zx. So why do we do that? We do that for two reasons. The first reason is that from this function, if you know the expectation, this expectation as a function of Z, you can have access to any moment of the distribution. The reason is just a property of the derivatives of the exponential. If I differentiate with Z, this, I get this. And if I take now Z equals 0, it gives me this. So it means that if I have the expectation of EZX, I can take a derivative of that expectation. I can put Z to 0, and I get, with this, I get the expectation of X. Basically, for example, I have the expectation of X is given by the derivative. I'm going to use the primes as I've done so far with respect to Z, taken at Z equals 0. But it goes beyond. I have actually all the raw moments with all the derivatives of any level. So that's why it's called a moment generating function. So if I take derivative to the kth level, kth derivative, first derivative, second, blah, 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 I get all the moments. If I know all the moments of a distribution, I know it's unique. So it means that this characterizes your distribution as much as a PDF. So if you play with the MGF and you find any result, it gives you information of the full distribution as much as if you played with a distribution function, with a PDF. The key thing we're using here, and we're using also it in demography and we're using it in evolution, is that MGF, moment generating functions, like most transforms, but this one is handy in our case, are tools that allow us to know what happens when you sum one variable with another variable. If I throw a dice 1 to 6 and I throw another dice 1 to 6, what is the distribution of the numbers I get? They go from 1 to 12. What is how likely it is that I get 6, 5, blah, blah, blah. If you want to know that, you can do it with the PDFs and you have to do sums everywhere or integrals everywhere. But if you do it through moment generating functions, it becomes often much simpler. And the important thing is here. Is it big enough on the screen that you can see?
Yes, it's visible.
Yeah. So after that, I will not do. I will get back to biology. It's just this is important, I think. Consider two variables, x and y. They are two different variables, whatever they are. And let's now consider the MGF of the sum of the variables. So for example, the count I get from the score I get from my two dice. So it's m of x plus y, and I take it at some parameter dummy variable z. So it's the expectation over x and y. I could write it x and y over the outcomes of the two variables of exponential z x plus y. And here I can write that as ezx times ezy. Now I do a critical assumption. I say x and y are happening independently. For example, my two dice, whatever happens to the first dice doesn't tell me what will happen to the other dice. They are rolling independently on the ground and they are doing their numbers independently. So here, then with independence, I can use a very classic property of expectation, which is the expectation of one variable times another variable that are independent is the product of their expectations. So I get a product of this expectation and this one. And what are those expectations? They are just the product of the MGFs. So the key thing is that when you're studying a variable that is the sum of many underlying variables, of several, whatever, many, don't need many, of several underlying variables, you get the MGF of this sum is going to be the product of the MGFs. Now, the Poisson distribution has an MGF. Some distributions don't have an MGF because they have some infinite moments. So not all distributions can be used with this tool. So I define a function here and I look for the MGF of the Poisson distribution. So you can see that it's an expectation of EXZ when X is distributed like a Poisson with parameter lambda. And that's the same thing. You can call it directly in Mathematica through moment generating function. And this function is a function of Z, it should be, and of the parameter of the distribution. And it takes this form, okay? The key thing we can have from that, which is the very important property of the Poisson, is a property that is the same as for the Gaussian. When you look at the sum of two draws from two Poisson distributions, the count you get from those two Poisson distributions is again a Poisson distribution, and the parameter of this Poisson is the sum of the rates of the two original distributions. And to see that, you can do it very easily because you say, here is expectation of x plus y. When x is taken in the first Poisson with rate parameter lambda x, and y is taken in another Poisson with parameter lambda y. And you can see that this is equal, because it says true when I say equal, it's equal to the MGF of a Poisson with parameter, the sum of the two lambdas. The reason is very simple. Huh? You can see it here. The MGF depends on the parameter lambda by just an exponential term. So when I'm going to do the product of the two, the exponential term will factor out and become lambda 1 plus lambda 2. it's just, but that means that you have with the Poisson what you have with the Gaussian. When you sum up two independent Poisson counts, you get a Poisson count. And you know the parameter of this Poisson count because it's the sum of the parameters of the two first ones. And of course, it goes on with as many Poisson terms you want. Another key property, so maybe I will skip that first. I will go to a direct consequence of that. You can look at a process now. If you look at a process, you're looking now at what is called the Poisson process. It's a process where things happen. It's a counting process. So it's counting how many things happen at a given time. And so say nothing happens, so process is at 0, then one hit happens, boom, it goes to +1. And then I wait, another hit happens, it goes to +2. And it's a count process, so it can only go up. And this Poisson process is defined by, there are independent numbers when you look at what happens from t to t plus s, and then you look at what happens from t plus s to, well, t plus s, and then from t plus s to t plus s plus q, two distinct chunks of time. The counts that are happening, the events that are happening in this chunk and the events in this chunk, they are independent. Typically, if you consider mutation, you will have things happening during the history of your population in a given part of the genome, and some hits happen, and it doesn't tell you anything about the hits that will happen in the other parts of the genome later on. So the mutation events happen independently over time. The other property is that it's describing a process where over some small time, you cannot do more than one occurrence. So over a very small time, there is not two hits happening together. Hits must happen a bit at different times. You cannot put two stuck together. It happens, but the probability of it becomes negligible as you narrow down the time where you're looking at. And the last thing is that the probability of an increment of +1 over some small time is just proportional to the time you're looking at. So it's lambda, the rate, times dt. And this process, when you have this process, then the increments that happen, this process is then a Poisson process, and it's called like this because the increments, the number of increments from time 0 to time t, is Poisson distributed with parameter lambda times the time for which you look at, lambda times t. So here is an example of what a Poisson process does. These are xt, the number of counts you have, and you see it's like steps going up and up and up, and these are replicates of the process. And it does this. So there's a trend, there's a variance around it, and it does jumps like that with various jumps. This one is very visible. You wait it for a long time, then one hit, and then another hit just afterwards, and wait again, etc. That thing will be the way we model mutations hitting the genome. And the reason for that is that, in fact, because we have said that the law of two Poisson distributions is a Poisson with rate, with parameter, the sum of the parameters. In fact, when we look over time, we can cumulate the Poisson counts from different times. And so we add them. It remains a Poisson because we have several Poisson and we're counting the sum of it. It remains Poisson and it's cumulating the rates over time. So it even goes on to a Poisson process that varies through time or space. For example, the rate, the intensity of the process may vary over time. It will be very important for us because when you have not many individuals, you have not many mutations happening in the population. But when you start to have more and more, there are more mutations that are fed into the population. So it's a Poisson process with a rate that changes as the population size changes. Well, in the Poisson case, and it's not true in any count process, you know that even if this changes over time, the number of hits at a given time, the increment over a given period, is just Poisson again, with rate equal to the integral of your rate over time. So you just integrate the rates that have occurred, whatever it does, going up, down, blah, blah, blah. You integrate it over the period you're looking at, and that's a Poisson again. And that's again due to the same thing we were saying. Two Poisson give a Poisson with rate sum of the rates. So it's a sum becomes an integral. And the same happens over space. So imagine there is a space, whatever space it is, some phenotypes, class, some ages, some actual space, and things occurring over this space, for example, the genome with different mutation rates at different places in the genome, then when you look at the whole process over the whole space, for example, over the whole genome, your number of counts is again a Poisson that is integrated over both the spatial domain, that I denote omega here, and the time. So this is very handy because we are staying in the Poisson with many complex spatial and time changes. Now, this is why Poisson is handy, but it doesn't tell us why we should use Poisson for modeling the mutation process. And the reason for that is in the fact that the Poisson has some kind of equivalent to the central limit theorem, but with the rare events. So it's well known that the Poisson distribution is a limit of the binomial distribution. And some of you have seen it, I guess, and some don't. So let's start with the binomial. In fact, when we think of mutation, we will typically consider that at a given locus, like at a given site in the genome, there can be a hit or no hit. So it's like throwing a coin tossing event. When I do heads, I get a mutation. When I don't, I get no mutation. So it's a 0-1 event. And then I have many sites. So the number of mutations happening over a gene or a genome is just the sum of all the hits that may have happened over those n sites. So in a sense, this mutation process is a binomial. The number is a binomial distribution. So the repetition of n Bernoulli variables. So there's a probability P of success for each locus, and there are n sites. So the number of mutations overall is distributed like a binomial. But now the problem is that we have multiple mechanisms. We have indels, so insertion of deletions. We have codon substitutions. We have transposon insertions. We have copy number change, like a gene, the whole gene is doubled. All these are different processes. They are different coins, and they are tossed with different probabilities of success. And on top of that, the number of coins that are tossed for each mechanism varies, because there are not all sites can undergo an indel, not all sites can undergo an insertion of a transposon, etc. So in fact, we're not dealing with one binomial model, which would be simple. We are dealing with a diversity of different binomials with different success probabilities and different number of draws. And that thing is very uncomfortable because the sum of two binomials is not a binomial. You can realize it by just considering just the sum of two, just one coin tossing, and the number of you get with two is not a binomial. So what we can do though, is that we can say that mutation is something that is rare on each site, because globally, most organisms, they have evolved to avoid mutation, because most of the time mutations does deleterious things. And so there's no organism known of where we have an estimate of the mutation rate, where this mutation rate per site is large compared to one. It's always small. Even, I mean, RNA viruses that are mutators, per site, it's small. And there are also many sites, because if you look even at one gene, there are many potential sites you can mutate. So we are typically in a limit of the binomial where we have many sites and a low success probability. So what happens to the binomial when you say that? So to model this, we're going to do one thing. We're going to say, okay, so that's just to show that there's no binomial equivalent to the sum of two binomials. We're going to say that we have a fixed mean number of things happening. So n times p, which is the expectation of the binomial, is going to be set fixed. But we're going to let n, the number of draws, go up. So mechanistically, p is going to go down. So we are keeping the mean number of counts fixed because we want a given rate of mutation in the genome. But we are increasing the number of sites from the binomial. And so we are mechanistically decreasing the probability that each site mutates. And if we do that, we can look at what happens to the moment generating function of the binomial distribution. Here is this moment generating function. It's given by this. I have reparameterized the P, the success probability per site, to the expected count divided by n, so that I keep a given fixed success rate over the genome. And now if I take a series Taylor series. So I won't explain what it is. You just ask your neighbor. A Taylor series of this, when n becomes large, what I retrieve is this thing becomes approximately equal to this. And this is exactly the form of dependence with Z that we have seen for the Poisson distribution. So that's how you can demonstrate that the binomial converges to the Poisson. What is important in this for our modeling is that while the binomial did not sum up to become a binomial, the Poisson does. So it means that with different mechanisms of mutation, we can pull them in as a single Poisson process. Because all the sum of the mutations, so if I call MI the counts of mutations of each type, indels, substitutions, transposer inserts, gene copy numbers, I want to know the whole set of mutations. They are given by the sum of those, and they are happening independently, considering that the process is independent of indels and of substitutions, which is not completely true, because if you saturate with mutations, the process cannot happen again and again on the same place. But as mutation is rare, you can ignore that typically. So then this thing, now, because we have this limit that it's not a binomial, these MI are not binomial, they are Poisson, then their sum is a Poisson. Then we can say it. And so it means that the mutation process is a Poisson. Even if we mix a whole lot of different mechanisms happening differently on different parts of the genome, we still get a Poisson as long as it's rare per site. So that's why it's not just comfortable. There's a reason to consider it. Just like there's a reason to consider a Gaussian for the distribution of breeding values when it's very polymorphic. And the reason is the same, it's just another limit of the binomial. So this is what I wanted to say about the Poisson. And I don't have time. I wonder if I have time to do the first ER scenario, but we're going to do it. We're going to do it very fast, and you'll let me know if you have problems. So I'm going to switch back to my board.
One question.
Okay.
Yeah, so since you mentioned that the lambda was time dependent, and also in some case it can be a space dependent. Can you elaborate that when it can be a space dependent, like in the evolutionary rescue problem? Or
in evolutionary rescue, we have, for example, when we start to look at mutation rates, mutations don't happen to the same thing every time. Some mutations produce a given growth rate, some others produce another growth rate. There is a distribution of the effects of the mutations. This is something we'll see after we have seen the simple rescue with a single type of resistance. If you want to go from your population genetics and rescue theory with a single type of resistance to multiple types, you use that because the space is the space of the growth rates. And the lambda will now be hitting different possible growth rates in the distribution of growth rates, for example.
Okay.
There's also, of course, cases where it's actually spatial. You could also think of it as a modeling the genetic mutation process at the sequence scale, as well. Space is the genome, and there are different rates of mutation at different places in the genome. But that doesn't mean you can't have a Poisson. What is important is that even if those things vary hugely between places, as long as you remain Poisson for all those places, then you have a Poisson for the total count.
Okay.
I can do a very quick overview of what we do for the first scenario of rescue, or I can say that we do this in the next lecture, because that was
Yeah, I think it would be nice to do some ER.
Okay.
Maybe we have about, I think, 5 minutes. So if you think you can do it, otherwise we can ask tomorrow. But it would be nice to do that today.
Okay. So I'll do 1 ER, and then I'll do the other ones at the next lecture. So the first ER we're going to do. So I'm still sharing this, and I should switch back to writing on the board. So the first ER we're going to do is this one. We have a stable population This is time, this is log n. We have a stable population. It's almost infinite. And there is a mutation rate, U, that makes you go from wild type to mutant. And this mutation has a cost C, which means that the Malthusian fitness of the mutant equals the Malthusian fitness of the wild type minus C. That's in the permissive conditions. In the stress condition, I'll put it with the red now. In the stress, the wild type changes fitness. It goes from some Malthusian fitness M. Sorry, I don't dare to erase. So Malthusian fitness M to some absolute growth rate R wild type. And this absolute growth rate is negative, so that the wild type goes down. And the mutant goes from its Malthusian fitness MM to some positive growth rate R like this. We are describing a bit more of this, so that would be R wild type. If we're considering cellular organisms, which is most of the use we have, it's a bit more relevant to consider that, well, it can be or not more relevant that we consider that mutation happens during divisions in this phase. In this phase, we have some rate U per unit time. The reason is that in stationary phase, where there is almost no division. So sometimes people say there is a bit of division and a bit of death compensating it. Some people say in other environments that in fact it's just a regular, it's getting, the genome is getting worn off. So there are errors, heritable errors coming in without divisions. But anyway, it's simpler to say there is a mutation rate per unit time because there's no division happening on really in the stationary phase. But here there are divisions, and most of the DNA is produced, copied, and a lot of the errors can be done at the moment of the copy, and they're heritable because they are copied. So then we would have a birth rate times a mutation rate. So that's the birth rate of the wild type, and that's the mutation rate, little u, per division. So first thing, sorry, my graph is wrong. My graph is wrong, but I wonder what's going to happen if I do that. So, okay, nothing bad happened, because I should be very high above that here, okay? I'm in a very large population. And then we're dropping at the onset of stress to some initial N0. Okay? Now, the way you can model what happens in this case, the key is to model how many mutations of resistant types are going to be produced during the permissive phase, and how many are going to be produced during the stress phase. During the permissive phase, what we say is that because we have a large initial population, we are going to have a mutation selection balance, because we say that Ni, an initial, this one, is very large. So we are at mutation selection balance. If we say we are at mutation selection balance with an asexual, then the frequency of the resistant mutant is S. In our case, S is minus C, because it's a difference between two mutation fitness. P, 1 minus P, plus mutation rate per unit time, times the frequency of the guys who mutate to the resistant, which is the other guys, so the wild type. Ooh. So 1 minus P. And we say that we've been in this state for a very long time, actually an infinite time. So because of that, we set to an equilibrium, P prime of T equals 0. And we have P equilibrium equals, if you simplify this, you will see, it's very easy to see, that it's equal to U over C. If C is bigger than U. Otherwise, P equilibrium equals 1. Well, this P equilibrium equals 1 is not relevant, and the costs are, if you're getting close to cost equals U, you have a mutation that is actually in the past, in the previous environment, that is actually neutral. So then you cannot deal with it by a deterministic model. You need to deal with it stochastically because there's no deterministic force happening on it, no selection against it. So we are really in this situation where there is a substantial cost and it's equilibrated. If there is no cost, then you need to model it with a stochastic. You cannot do a mutation selection balance because there's no selection. You do a drift mutation balance. And if the cost is negative, which means the mutation is beneficial in the past already, then there's no issue of rescue because basically your mutation will invade before the stress and the stress will happen and the population will not see anything because it will have invaded the resistance already. So we're in the only situation where we can use a deterministic model and it makes sense to wonder about rescue. The mutation is deleterious in the past. Now, from that, we know that in our population, before the bottleneck, we have an equilibrium U over C. After that, we do a bottleneck. To reach size N0. So what happens in this bottleneck? Basically, the number of mutation MSV, I call it. It's the number of resistant mutation at onset of stress. This number is really a binomial. N0, P equilibrium, because you're sampling N0 individuals in a population that is infinitely large, so with replacement, and that has a fraction P equilibrium of rescue mutations, of resistance mutations, okay? So it means that it's a binomial. But then you're saying that P equilibrium is very small compared to 1, and that N0 is large. So we're dealing with a finite but large population. And that means that you end up with the Poisson limit we just saw. The number of mutations of rescue is Poisson with rate, the mean, which is N0 P. This is the number of resistant mutations. Now, a rescue is not a resistant mutation. A rescue means resistant plus established in the future, meaning it will establish, those mutations we get, some of them will be lost, some of them will establish. And by establishing, we mean avoid stochastic loss when rare. And this happens with some probability, PE. This probability is fixed for the mutant we are considering. It's due to its demographic properties, its birth and death rates, its say its age, its lifetime distribution, potentially many things, many details of its biology. But it's a fixed thing, a fixed property of a genotype from one copy. Rescue is going to be the number of rescue is going to be the number of standing variant resistance conditional on establishing. And there we use a last property that I didn't have time to tell you about, the Poisson process, which is that when you condition your Poisson process to some Bernoulli thing, happen or not, some color, you still have a Poisson process, but the rate is multiplied by the probability of this success. This is just also true in the binomial, actually. You say a coin tossing and plus my coin fell out of the table. And I'm considering only the coin toss that fell on the heads, plus they fell on the table. Well, then this is multiplying the probability to fall out of the table times the probability to do the heads. So it means that MSV star, which is the rescue, is going to be Poisson with rate, again Poisson, with rate N0, PX, PE. So we can condition the numbers from the past by what they will do in the future, because we're giving a fate to each mutation for the future when we're counting them right now at the onset of stress. We're saying, okay, among all of you guys, we know that there is this bunch that will die out. Goodbye. But I don't have, you've not died, but I know there is a probability of this that you will die. So I can already count them. So if I do that, I have corrected for the fact that of all those guys present in the population, a subset will be lost by stochastic loss. And then you have an explicit expression for the number of rescue in the standing variance. It's N0, P establishment, mutation rate over cost. Now, we are looking at the second phase in the stress. We'll use the two results after that. In the stress, we have a decaying population of wild types. It decays, so it has a size n of t, and it decays because we are having a density-independent growth as N0 E minus RT. Sorry, it's like this. I will keep my notation. E R wild type T. And R wild type is negative. It's the slope of the log n decay. Rigorously, it is. It's measurable this way. So this is our wild type. And the population, because we're assuming that U is very small and that the mutation rate after the onset of stress is also very small, B times U, then most of the production of mutants comes from the wild type all along to the end. So we can model the mutation process just for the wild types producing mutants. And what we have then is that from T to T plus dt, a small chunk of time. During this small chunk of time, the number of mutation of rescue is what? It's distributed as a Poisson with rate n of t, this, times the birth rate of the wild type times the mutation rate per division, dt. I'm not writing it right because rigorously it is a time inhomogeneous Poisson process with rate changing proportionally to the pop size. Because of the properties of the Poisson that we saw, sorry, I have to add this. These are the numbers of resistance, not rescue. This thing means that I'm having a Poisson process with inhomogeneous rates. And so it means that the number of guys I get is a Poisson. The number of hits, be careful, huh? It's the number of hits I get. So the number of rescue mutations that arrive. So this is the number of rescue mutations that come into existence. It's not their number.
Guillaume, sorry to bother, sorry to disturb, but I think we're already 10 minutes over time. So shall we stop here and take it from here, perhaps?
Yeah, okay. Yeah, I've done the standing variants already. We'll do it. We'll take. Yeah. Tomorrow we'll do this ER scenarios.
Right. One suggestion, maybe if you find it more useful, maybe you can try to use this Jamboard. I don't know if you're more comfortable with that in terms of erasing.
What is the Jamboard?
Jamboard from Google Chrome, you can get it.
Okay. Yeah, I'm going to
just try because just I find it easy and erasing and all that stuff is not a problem. But just try this.
Okay, I'll do a try with this there. Yeah.
Okay, so
because when we did trials, it didn't show up this problem of erasing.
I don't know. Yeah, I don't know. Anyway, yeah. So we'll see you tomorrow.
Should I put the notebook on Slack already, or
you can put that
for tomorrow?
Yeah.
I put it for tomorrow already.
Okay. Yeah.
Because it's gonna retrace what I've said about the Poisson and derive the rescue models. So.
Sure. Yeah. Okay. Yeah. See you tomorrow.
Okay.
Bye.
See you tomorrow.